Because there are a lot of existing K27 acetylation, ChIP-seq, and chromatin accessibility profiles already generated in the public, sometimes, even if you only have differential genes without any TF ChIP-seq, histomark ChIP-seq, or DNA ChIP-seq, we have an algorithm developed, we call it LISA, that can already tell you which transcription factors might be important by utilizing the public TF ChIP-seq and histomark ChIP-seq or DNase data. And so the approach works like this. The algorithm takes an input of, like, say 100 or 200 differential genes between some conditions. And based on those differential genes, the algorithm first look at all the public K27 acetylation or DNase data, and use each data to calculate this regulator potential. And so you can see in this case, every column is one DNase or one K27 acetylation ChIP-seq data. And some genes, where we calculate this for every gene, so every gene has a score. And so some genes are high and other genes are low. And these are probably the active genes in the cell. And these are the not active genes in the cell. And so we first ran a regression model to ask if we need to have a kind of we collect, say, the top 10 K27 or DNase profiles using a lasso regression so that the top genes agree with the differential input gene that the query gene list. And so say these 10 are being selected, and we know that using lasso regression, these 10 together will give us the genes, their RPs are very different from the rest based on the K27 or chromatin accessibility profiles. So we say, okay, these are the ones that we select. And then together, these epigenetic profiles, because some of them might be K27 acetylation, some of them might be ATAC-seq or DNase, they tell us which regions are important for enhancers. Then we look at whether it's a TF ChIP-seq in the public or the TF motifs. If we don't have the ChIP-seq data, we ask, okay, which TF ChIP-seq really bind to these enhancer locations that have the K27 signal or the DNase accessibility signals. And then we try to see if you were to not have this TF ChIP-seq or the motif, how much does that change the nearby genes? And we don't just do this in one, we do it across the genome of all these K27 regions or DNase regions. And we ask, how much does it change the nearby gene? And the TF that change as most of the nearby genes as your query gene, that's your TF of interest. And so basically using this, the LISA approach is, we first try to use public epigenetic data to select the relevant one that indicate this particular cell type. And using that epigenetic profile overlapping with the TF ChIP-seq or motif occurrence, we can evaluate which of this TF effect has strong influence on the nearby gene expression. And then we'll figure out which factors might be important. And so you can imagine if you compare the differential genes between conditions, like there are some upregulated genes and there are some downregulated genes, you can run this through LISA and it will actually tell you, which transcription factor is important for upregulation, what other transcription factor might be important for downregulation, and there might be some factors that influence both, right? And so you can see here, Cystron Toolkit, if you have just the ATAC-seq data or the DNase data or histomark ChIP-seq, Cystron Toolkit can find the TF ChIP-seq that overlap with your ATAC-seq. But if you don't have any of this at all, you only have differential genes, you can still run LISA and use the public ATAC-seq or histomark data to help you make predictions. And they both kind of use the TF data in the public as well, which is informative.